Welcome back. Thanks. We'll get started with our second short talk session. Our first speaker is Mercedeh presenting PathSeeker, a statistical package for enhanced pathogen identification and characterization in RNA sequencing data.
Hi, everyone. So I'm very excited to be here, and I'm going to discuss with you our new package, PathSeeker. So first off, I guess you guys might want to ask, why do you even use RNA-seq for calling pathogens? Well, there are various reasons. You may not want to be limited to bacterial organisms. So when you do RNA-seq analysis, you can identify pathogens that are of viral, fungal, parasitic, phage, or bacterial origins. Generally speaking, with RNA-seq, you're dealing with RNA, which is less stable than DNA. And a lot of the times, because DNA is everywhere, you get a lot of DNA contamination. So methods like metagenomics are also, they also do pick up these pathogens. However, contamination is really high. RNA provides a unique feature, and that is it shows you active infections in comparison to DNA, which might be remnants of a prior infection. And finally, when you're doing RNA-seq and you're identifying pathogens through this method, what you can do is combine pathway signatures from the same patients and see if there's an actual immune signature happening in these patients that you identify pathogens in. And so what is the problem? Why do we need to develop a package for this? As you can see, I mean, you could use various tools to just identify pathogens. They mainly, what they do is they perform alignments and they generate taxa tables like this. This is an example of a taxa table created by CZID, which is my favorite method for identifying pathogens. And you can see your x-axis is actually the samples, the y-axis is the bacteria, and this is like 1/10 of the whole heat map. So basically you get a huge heat map that is all red. And so the challenge remains, what is the actual pathogen driving the disease and what is active in these patients? And so that's where PathSeeker comes in. What we do is, so PathSeeker is a combination of various statistical methods to call pathogens for any sort of, through any sort of RNA-seq data set. And so we look at it in two different ways. One is the standard one that you see right here to your right, which is basically differential abundance or expression in that matter, which also applies. And you could use non-parametric tests or model distributions. So we have wrappers that actually you could use if you use PathSeeker that basically make your data set translatable to these methods. For that, though, you have to have diseases where there's clear good number of pathogens that have caused a disease in your data set that are of same origin or like same community. However, in reality, most of the diseases that I work with and a lot of infectious disease doctors work with are like neonatal sepsis and infectious hydrocephalus. They could be caused by many pathogens. So in a data set of 100 samples, you can have 40 different organisms leading to infectious hydrocephalus or neonatal sepsis. So the differential expression models at that point don't really have enough power to pick up what's going on. So what we recommend in those cases is basically doing simple steps and some statistical modeling for calling contaminant organisms, followed by other sort of soft filtering, and then identify your organisms and then do visualization, which is all possible through PathSeeker. So what do I mean by that? I'll give you a brief introduction of decontam, although it's not something that we have developed for PathSeeker, but the package does support it, and it gives you a little bit of background on how to look at this. Generally speaking, when looking at pathogen calls, samples that have good quality, have high DNA concentration, show less contaminants. And the other way goes around. So if you have a sample that has bad quality, it will show high levels of contaminants. And so this package uses log transfer linear models to identify what is a true organism versus a contaminant organism. In PathSeeker, we're proposing using topic models, and a brief introduction to how we're using our adjusting topic models to call contaminant versus true organisms. You're dealing basically with a bag of biological words where your words are the taxa and the samples are your documents. You identify the optimal number of clusters. The package does provide still a vet method for identifying optimal number of clusters. However, you can use your own method and you can just input what is the optimal number of clusters. The data gets classified clusters, and what you get out of that is basically two data frames. One of them is it's the theta, which basically shows you the weight of every feature in every topic, which is represented here. The other one is omega, which shows you the weight of the sample in that topic. You can look at this, that should explain it. So basically, this is your theta, where it shows you the weight of your features in every topic, and each of these are topics. And for your omega right here to the left, each of these lines is a sample, and the colors represent the clusters or the topics, and it shows you how much of that particular sample has that topic. So if you think about it, if you assume that your sample set has a baseline, what do I mean by baseline? I mean blanks or like experimental controls. Those can act as your baseline baseline for contaminant topics. So what you do then is to measure the weight for that blank topic, basically see which topic has the highest level in that blank sample. And then those features are highly likely to be your contaminant features. So then what do? You go back to your whole data sets, so your theta right here, and you see which one of these features have the highest level in that particular topic, and those are the features that are likely contaminants. You tag those, and then if you want to remove them, you can remove it, or if you do any sort of other forms of analysis, you can always keep in your mind that these are potentially contaminant taxa. Another way which really it helps to think about this is if you do not have any baseline, basically if you do not have any controls for your analysis. In that matter, as I discussed with you, samples who have high abundance and high prevalence most likely are the samples that will show you more of your contaminant taxa. So you are trying to select these features. And once you do that, now that you have that theta that shows you basically the weight of every feature and shows you which ones have more weight in that topic, you sum that up and you figure out through your theta which topic is potentially your contaminant topic. And once you do that, you sum, basically the sum of that contaminant topic needs to have a threshold, because if you sort of have a little bit more in one topic than the other ones, that's not really fair. So it has to have at least one time more, the contaminants have to be at least one time more in that topic than the other topics. And that's also adjustable. If you want more strict, you could do more strict cutoffs, and then once you do that, you go back to your data set, you measure which topic you get, which one of these features has the highest level in that topic, and you eliminate accordingly. And so after that, you could do soft filtering, and followed by, the package has a lot of plotting and heat map and visualization things. We have looked at various data sets, and also we have gold standards, which are qPCR, and it seems to, for different types of organisms, it seems to work really well. And so, for example, for Plasmodium, we've checked both qPCR and rapid blood tests, and it matches really well for bacterial organisms as well, or these could be prior contaminants that it does not pick up, and also for viral organisms. Finally, just quickly, RNA-seq can be used to identify live and active infections, and this package uses various forms of statistical and machine learning methods, and it's easy to use, so clinicians can also use it. And hopefully, I hope by tonight or tomorrow, I'll submit it to Bioconductor. Thank you.
Lovely. You always have something new every single time. So the bag of words for microbiome or how to put it, metagenome decontamination. Have you thought of calling it the bag of bugs or something like that?
The bag of contaminant bugs.
There we go. Bag of
Most of it,
contaminants.
not all of it.
So I noticed that you did some gold standard benchmarking for CMV reactivation.
Yes.
And what was the method by which the metagenome was assembled for the being compared against the qPCR primed or the PCR primed CMV?
So this is all like RNA-seq data. So the calls by PathSeeker for RNA-seq being positive versus qPCR. So I think you're getting to the point that why is like some of the qPCR like not positive while RNA-seq is positive, and that's because it's targeted when you do qPCR, and we know CMV has a lot of non-coding regions, and so probably RNA-seq is picking those up, and the qPCR just won't.
So a follow-up, is that from short-read RNA-seq then?
It's possible. So when you throw the calls are made from short or long read, so anything with 150. So we do 150 base pair for RNA-seq, but it will still pick up shorter reads. So you could pick up microRNAs and other things with RNA-seq as well.
So I was sort of aiming at was if you were to do this standardized against, say, long read, say Nanopore, PacBio, would you expect a tighter concordance between them?
We actually talked about this. I talked with someone, he's sitting in the morning, about using Nanopore. So yes, that is a thing that we want to do. We want to do real-time calls for using RNA-seq to see active infections. I haven't done that yet, but it could be benchmarked that way as well. Yes.
Really cool project. Thank you.
Sure. Thanks.
I know there were some more questions for. Maybe you guys can find her at a break or a lunch. Just moving on for time. Our next speaker is virtual. Talk is pre-recorded, but they are in the chat room if you guys are on the platform. So they are there and they will be here to answer questions live.
Good afternoon, everyone. It's my great pleasure today to give you a short talk about one of our recent stories on AMR, an R package to predict antimicrobial resistance features in bacterial pathogens. I'd like to thank the Bioconductor organizers for inviting us here. So in our group, we love all things related to computational biology. We especially love microbial genotypes, phenotypes, and diseases. We're also deeply passionate about education, outreach, and diversity in data science. We develop computational methods that span multiple molecular scales. Few of these on the pathogen side, and few of these on how the host responds to these bugs and how can we treat them. And today I'll focus on antimicrobial resistance, which no doubt all of you have heard about. This is the One Health issue, which means it affects humans, animals, and the environment. So in 2019, the cases seem to have gone down a little bit, but then, COVID hit. So even in 2020, early 2020, there were close to 3 million infections each year and more than 35,000 deaths purely from antimicrobial resistance. So this work was started by a few undergrads in the group and most recently led by Jake, Ethan, and Evan. So I'll tell you a little bit about each of these, but I just wanted to tell you we have a work, an R package in progress. So there is a page right now, but it's still private and very soon it will be public. The input here is pathogen genome sequences, the corresponding isolate metadata, and the antimicrobial resistance phenotypes that go with it. And what you would get out of the package is a bunch of machine learning models that you can run on these data. You can subset it by bugs and drugs, a whole bunch of data summarizations and visualizations, and most importantly, the top AMR genes or the features that are giving rise to these predictions. So for today's talk and for the initial part of this package, we are going to focus on genes because they are among the most interpretable of these molecular scales. The kind of data that we have is a whole bunch of genome sequences, which you can abstract in this form. of a matrix, if you construct a pangenome, it basically will bring it down to a genome by gene matrix. Per genome, per isolate is a particular gene cluster present or absent. And then we also have the phenotypes. In this case, what this means is whether a particular genome is resistant or susceptible to a particular drug, meaning does it respond to it or does it not? Does it kill the bacteria or not? And this is the abstracted version of the data, and this is going to be our starting point for our machine learning models. And we train one model initially per bug and drug combination. So we download a whole bunch of pathogen genomes, anywhere from a few thousands to several. We annotate every one of these genomes to give us genes and proteins, and then we construct these gene or protein clusters. And then we remove any genomes that don't have phenotypes initially, because that's important for the training part of it. And then we also remove genes or proteins that are present in pretty much all the genomes or pretty much none of the genomes, because we think it's unlikely that they're giving rise to this resistance of susceptibility phenotype. And these feature matrices are the input to our machine learning models. So the brief overview of the machine learning workflow is you have a feature matrix like I just showed you. We have labels, we binarize it, again, just for initial ease of interpretation, but very soon we'll also take into account the concentrations of these drugs. We build a whole bunch of classic machine learning models. We start with logistic regression, but we've also run random forest, SVM, and a few others, including boosting algorithms. And we start with these because they return probabilities, they're interpretable, we know what are the features that are giving rise to this prediction, and that's the reason. So let's take an example. This is Staph aureus, a notorious pathogen, and we're looking at penicillin, which is one of the classic drugs. So initially, this graph here shows the class balance, which is to say how many genomes we have for the resistant and for the susceptible phenotype. And what we have here is the AUPRC, which is the area under the precision recall curve, which is a proxy to say how well is the model performing when it comes to this prediction task, or the classification task more specifically. And this was remarkably good, right? 0.99. So we immediately wanted to see what are the features that are giving rise to such good performance. And we were pleasantly surprised to see a lot of recognizable genes. Some of these blah, blah, blah genes that you see are all beta-lactam resistance, which is basically the drug class that penicillin belongs to. So that was a good proof of concept for us. And MEK is, again, part of the methicillin resistance, which is part of the penicillin class of drugs. More interestingly, we found a whole bunch of these top genes are all implicated in horizontal gene transfer. For those of you who are not biologists, this is basically part of plasmids, which are mobile genetic elements. These are genes that are moving around from one bug to another. Yeah, it is exactly as dangerous as it sounds, which means if one of these bugs has learned how to get resistant, then very quickly other co-inhabiting bugs can get these plasmids too and become resistant to these drugs. And we find that even with drugs like tetracycline, where there is very, very poor class balance, we still seem to be doing very well. And again, one of the top features is the interpretable TET-K, which is involved in tetracycline resistance. So we run these models similarly for a whole bunch of drugs in Staph aureus, and they seem to almost always be doing very well. We also run these for Klebsiella and a whole bunch of these other escape pathogens. So this is a brief summary to show that we've run it for a whole bunch of bugs, which is these 6 pathogens and anywhere from 5 to 15 drugs per bug. And the current functionality of our package is you'll be able to load all of this genome data and metadata for these AMR pathogens, starting with escape. We also have modules or functions that you can run for multiple machine learning models based on how your data is structured and which ones you want to run and compare. And we also give you the performance metrics for each of these. And you also get the prediction of the top genes per bug drug combination. There are also a bunch of functions that will help you with getting summary stats and visualization for the AMR metadata, and also for your final predictions that come out of the machine learning model. What's next? So one of the things that we're very interested in is to see not just the static snapshots of which bugs and drugs are resistant, but to see what is the evolutionary context. Where else are these genes present, and how have these mechanisms evolved? And we clearly want to escape this beyond escape pathogens, but this was a good use case because they are notorious for AMR. And we're also now working on predicting cross-resistance across bugs and drug families. And all of these will become functions in our package too. And currently we are focusing on genes, but we would also like to go to other features. And we're also adding a Xinyue dashboard in case that's an easier way to visualize when you're looking across these multiple comparisons. And all of these will be linked out to the corresponding resources for the biologists who are interested in the downstream outcomes. And we hope to have a beta version for you all to test out in a few months. With that, I would like to thank our fantastic group and stay tuned for Raymond's talk, which comes right after this. I also work very closely with the R community, and I want to give a huge shout out to them and our funding sources. And with that, I'm happy to take any questions. And don't forget to, if you're an R Ladies or an R Ladies ally, please join our lunch tomorrow at 12:45 p.m. and find Stephanie Hickey to do that. Thank you so much.
We do have Dr. Ravi virtually, if anyone has any questions. And nothing in the group.
I see one question on chat. Can I answer that?
Yep. Can you read the question out loud for us?
Yes. Svetlana asked, have you tested other pathogens beside escape? The answer is yes. We are starting with other bugs beyond escape. For instance, mycobacteria, whether they're MTB or non-tuberculous mycobacteria, or they have a lot of AMR features. But that's where we want to go beyond genes, because genes themselves are not sufficient in predicting AMR in some of these pathogens. We have to go to these other scales like SNPs, because that's where the genetic variation comes from. But yes, we are testing other pathogens beyond escape.
Well, thank you so much. I'd like to welcome our next speaker, Raymond Lesiyon from Michigan State University, with a microgenomeR package, an R data package for integrating genomic metadata and bacterial phenotypes. Thanks.
Hello, my name is Raymond Lesiyon. I'm from both Michigan State University and also CU Anschutz Medical Campus. I'll be talking about microgenomeR, a package for integrating different genomic data sets. So the main motivation for this is that there was a project we were working on that we wanted to understand which kind of traits do we differentiate between pathogenic bacteria from other bacteria. Most scientists do study this on a like a micro scale by comparing between strains within a species. We wanted to like compare this for like all, like let's say for all on a bigger scale, are we able to pick traits that distinguishes a pathogenic bacteria from others? So to do this, we require access to really large data set and also like a high quality microbial data to be able to answer this kind of question. So there are like some data set already available. So like, for example, this is called Made in 2020. This was collected in 2020, but it's a significant focus on marine organisms. There's also bug dive, which is limited to cultured organisms. And you can see, so these are like how the data set looks like. And there's a bug fish, which combines around like 17 different data sets. And this was like done by a Worldry lab in Kune. So why do we need like a new data set apart from just using the one which is already available? So some of the data sets are really, most of them are like in PDF. So sometimes you have to copy them to Excel, or you have to scrape them using scraping languages. Those are available in databases. You really have to fully access them. You have to use an API. Additionally, some like Made in 2020 and Bugfish, they are really big missing values. They're like really big missing values. And also, like if you're interested in other kind of traits, you'll have to, as some of these data don't have this kind of traits. So we wanted to use microgenomeR to make this data set readily available to people by either an R data package or a packet. We also wanted to clean this and process this for people to readily use it. We also wanted to ensure that this data set, someone can easily analyze them using an interactive dashboard. So how do we do this? So we combine a lot of data sets, like around 20 data sets, and each data set will have like a respective thing that we get from it. Like for example, BVBRC, we get genome metadata and the AMR phenotypes. For NCBI, we get genome metadata also, but with assembly stats. And for Fibase, we get like a list of curated pathogens with the OS interaction. So how do we do this? We repurpose a really nice workflow established by Osrate, a curated plant data set that enables you to like harmonize a lot of data set together. So how do we do this? We start with like a bunch of data sets, then we process it in two formats. So harmonize it and also allow traits. Harmonizing means we ensure that they have the same column names. We also ensure that they have the same trait values and also ensure that they have the same units for a particular data set. Then from that, we also make sure that things are in allowed ranges. Like for example, GC content, if like in the literature, they're between 15 and 17, 75, we want to make sure that they are in that particular range. So at this level, then we're able to clean all different data sets into a really nice format in which we can now combine them into strain level or species level and put it as a form of .rmd or packet that someone can easily load. So with the data set, someone can answer like different questions and be able to access the data set easily. For example, you can answer how does GC content change with oxygen requirement? So to do this, you first need to load the data set, which will now be locally available, easily available. So you have to load in either data level. So this is like either species or strain level. Then you can also like load them in a particular taxa. So you can either use all the taxa or like use a specific taxa. I will show that in a bit. And the columns that you need. Then there will be functions that will enable you to like make different plots. In this case, you can make like you can make a box plot. You can make a box plot by a different numeric GC content faceted by oxygen requirement. And this you should be able to use any category of your interest. Additionally, you can do the same thing, like let's say, how does GC content change the growth temperature? You load, so the same thing, strain level or taxa level. In this case now, taxa level, you want a phylum. In the phylum, you can specify which specific phylum you want, and you'll be able to visualize your data set. So in summary, we were able to like combine around 7 data sets. There's a plan to, we're planning to extend this to like around 20 data sets. This kind of harmonized data set will enable someone to do like a macro scale analysis, like a phylogenetic regression between different traits. And you can perform this either in a strain level, species level, or particular taxa level, just by the way you subset your data. Going forward, we are planning to incorporate incorporate the visualization I just showed below into an R-Shiny dashboard with customized functions that someone can easily use to do the analysis. We're also planning to collaborate with a Wardred lab, which they develop to develop a large microbial phenotype. They already have something called Bugfist that we will incorporate into ours and like make a big a microbial phenotype repository. Additionally, because it's a lot of harmonization we have to do, we want to use a natural language processing or large language modeling to be able to harmonize different unstructured text in a much reproducible way, or much easier way. We also want to link this to either like be able to link different genome metadata, different genomes to their the respective metadata. With that, I would like to acknowledge my mentors, Dr. Janani and Dr. Nina, and also you can look at their labs, and also you can have a look You can have a look at the package over there. Thank you.
Sure, nobody else has any LLM questions? So I recently served on a panel with Philip Payne, and his group at WashU uses LLMs and similar machine learning approaches to extract information from unstructured fields in electronic health records. And he pointed out that oftentimes the most interesting information or the most useful and informative aspects of many patients' histories is unstructured in notes. So I'm curious for the aim of this project, how you are planning to implement and benchmark extraction of unstructured data for microbial communities and microbial resistance.
Okay, so the way we're thinking of there's for different data set, they have the names for a particular trait are really different. So we want to use first we're trying to harmonize those different trait columns to be sure that they're in the same format for all the other data sets. So that would be one easy way for us to validate that. Additionally, for other bacterial traits, so what we're thinking of using is something like similarities. So for if we have a particular text and we want to see how similar is that text to a particular metadata that we're looking for, and I'll probably validate that manually before expanding it to do in a large throughput.
Cool. Well, I'll be excited to see the results.
Okay, thank you.
Our next speaker is virtual as well. They are giving a talk on inclusive internships in genomic data. Nope, whoops. Okay, good. Got the right one. Inclusive internships in genomic data science, Outreachy and Bioconductor full screen. By Svetlana Perovic. Hopefully, I probably butchered that name as well, unfortunately. But thank you so much. And I believe you are also online, so people who have questions can put in the chat, and people can ask questions after, and she can answer live.
I'm Svetlana. I'm a microbiome scientist from University of Trento in Italy, and I have had the incredible privilege of mentoring outreach interns over the last 2 rounds. Today, I'm excited to share with you our journey in creating inclusive internships in genomic data science, focusing on the synergy between Outreachy and Bioconductor. There is a critical issue in our field, the inequitable access to modern genome scale biology tools and methods. To address this, we have partnered with Outreachy, and they are providing internships to underrepresented groups in tech. Together, we are creating internship opportunities in software development and genomic data analysis. This collaboration aims to open doors for talented individuals who might otherwise be excluded from these cutting-edge fields. Let's look at the timeline of our Outreachy Bioconductor projects. We have had 4 rounds of internships so far, each focusing on different aspects of bioinformatics. Our first round was in December 2022, involved 2 projects, SWIV 2RMD and BS Genome Forge. We had 2 interns, Ber from Kenya and Kirabo from Uganda. The second round in May 2023 expanded our scope. We continued with SWIV 2RMD, added Miaverse and BacSigDB. 3 interns, Sonali and Pratheepa from India and Dina from Cameroon, were mentored by Jen, Thomas, and Chloe. The last two rounds focuses on BacSeq-DB, and we have 4 interns, Peace, Xinyue, and Esther from Nigeria, and Scholastic also from Nigeria. And they are being mentored by Chloe Levi and myself. What about this timeline? So this timeline showcases the growth of our program, the diversity of our interns, the range of projects we are undertaking, and it's exciting to see how we are gradually increasing our impact and reaching out to talents from various countries, particularly in Africa and Asia. Moving on to our evaluation efforts, we conducted a survey to get a feedback. This is a crucial step for us in understanding the impact of our program and identifying areas for improving. In May 2024, we distributed an anonymous online questionnaire to all applicants who contributed to our Outreachy Bioconductor project across all 4 rounds. This included over 200 individuals who engaged with the various projects. I'm pleased to report that we received 59 responses to our survey. We can see here the geographical distribution of our applicants across 10 countries, strong African representation, significant participation from India. And this diversity aligns with our goal, increasing access to genomic data science in underrepresented regions. But moving forward, we might consider strategies to broaden our reach in other countries and regions to further diversify our applicant pool. The high percentage of female applicants, 85%, almost 85%, show strong interest from women. Diversity of applicants, including students, freelancers, currently unemployed, indicates that the program's broad appeal and potential for impact across different career stages. The statistics and testimonials reinforce the importance and positive impact of our internship initiatives, and especially in empowering women in scientific computing fields. Further, we observed the impressive growth of our internship program in terms of number of applicants. Also, we expanded the bioconductors projects, and in the end focused on BacSeq-DB as a major project that we are offering right now. Here, I would like to highlight the increasing interest in our internships, the last 2 rounds from December 2023 and May 2024. We had an impressive number of contributors, mostly from Nigeria, with a significant representation from other countries, India, Uganda, Kenya, and among others. During 1 month of contribution phase plus 3 months of internship, our contributors and interns have made tremendous contributions. For example, the number of studies created has grown significantly from 800 in March 2024 till today, more than 1,000 curated studies. This growth underscores the importance and impact of inclusive internships in genomic data science. Beyond the internship, we see ongoing engagement from our participants. 6 applicants from December 2023 returned to the next round. For the next round of application for the internships, interns Peace, Xinyue, and Esther stayed with us in the community as commentors. Additionally, 10 applicants from the May 2024 round continue to contribute. And we are also seeing increased online activity with the applicants sharing their experience through social media, blogging, at the conferences. This sustained involvement underscores the lasting impact of our inclusive internships in building a vibrant and engaged community. For the next internship round, you are more than welcome to join us. If you are interested in mentoring, please reach out to Jen for more information. Here are listed the key dates. We encourage you to join us and to continue together fostering growth and development within the bioconductor community. Finally, I would like to thank all the people who have been supporting this work and initiative: Sagata Lab, Valdor Lab, BacSeeker team, Outreachy team, and special thanks to my colleagues, Jen, Maria, Chloe, and Levi, and of course, all the past and current bioconductor mentors and contributors. Thank you.
Hello there. Thank you so much. Really wonderful presentation. And I think a tremendous amount of work to assemble information about what's being done. I think one of the most striking things is this notion of co-mentoring by individuals who were interns in the previous period. So this idea of train the trainer, I think, is kind of fundamental to what these programs are trying to accomplish. How can you capture at the right level of detail information about how this is propagating and how trainers are training trainers that become trainers and so on? Do you have a database or some mechanism for recording this that isn't so, I don't know, manually intensive as what you must have done thus far?
Yeah, thank you. This is a great question. So yeah, we are really proud of our commenters, and yeah, actually in the BacSeeker, so the last two projects that we are offering in the Outreachy, you can record the contribution of each person, and our previous interns, and their contribution was so huge, and not only huge but also discussion in the Slack, and their efforts to bring like team contribution even and their effort as a great example to get them as a trainer for the next round. So this is actually also the Outreachy. One of the Outreachy's goals is to keep the interns in the community and to expand the Outreachy activities in that way. So we are really looking forward, for example, from this round, which is ongoing, to have also more trainers with us. Thank you.
Yes. You can. Okay. All right. So yeah, thanks. Also, like, this kind of talks about the community. That's what I like about the Bioconductor conference. One of the things that I like about this conference. So yeah, also like a great job in doing the outreach in Africa and India. So kind of have one question and one comment. So one question. So it's about the students from Africa, India, and so on. They will have very different cultures from you. So what do you think you have learned from their culture and their perspective, and also like how does the content of the internship be? How would you make the content of like that internship like culturally relevant to them?
Well, this is really great question. I learned a lot from them. First, to be patient, and second, to develop more our for example our instructions for doing like different tasks, and
yes, some things that we skipped or we know by default. They pointed out that we should put etc. But okay, this is not so cultural. But yes, like everyday things that especially in scheduling the team meetings or in the discussion of different things, not necessarily connected with the project, but with open source in general. Yeah, we learned a lot.
So like how specifically do like the content of the project more culturally relevant to them?
Sorry, could you please repeat it?
So like how did you like make the content of their projects more culturally relevant to them?
This is something that we will work on it, because as the previous question was highlighted, that we train the trainers, so we are thinking to join them in our team and make these things better, so more fitting to their culture.
Yeah, so
we're working on that.
Yeah, that's a great answer. So next I have a comment for, looks like you really have like an international focus, but meanwhile I think it could be a good idea to do it more locally as well, because like even within the developed countries, there say LA, I lived in LA for 11 years, and I know that like there are many underserved communities in LA, like like Beverly Hills is very rich, but we also have South Central, we also have Watts, Boyle Heights, East LA, like those are very underserved and they have low income, and yeah, I feel like it would be great to do more of the outreach like locally.
I see. Okay, yeah. I mean, great suggestion. So we definitely should work on it.
Thanks. Great, thank you.
We will have to move on for time, so I'm sorry with the last question, but maybe you guys can reach out in the Slack for the conference if you guys haven't already joined the.
Definitely, I would like to. Yeah, definitely.
We have a lot of interest in the room. You can't see it. But thank you so much.
Thanks.
Our last speaker for the session is also joining us virtually. There are two co-authors on this talk. I don't know if any of them are present virtually to answer questions after, but I will try to find that out while they're speaking if they joined. I know they weren't originally logged in, but maybe they've joined since. This is Statistical Methods for the Tissue Microenvironments by Lorraine Chu.
Hello everyone, I'm Xinyue. Today I'm going to talk about the statistical method for tissue microenvironments. I will provide some background, discuss tools for data exploration, introduce spatial latent directly allocation, which is the LDA, and then proceed the model diagnostic and the model assessment. For some background about the multiplex L-beam imaging time-of-flight data, the MIBI-TOF technology generates spatial data across multiple samples and providing data that includes various protein expressions in cells and their spatial coordinates. By clustering these expressions, we can identify different cell types as shown in the figure here. Different colors represent different cell types. And then we perform spatial partitions based on cell types to reflect the tumor microenvironments. The well-existing spectral LDA technologies do partition images based on the cell types, but are somewhat limited in identifying partitions due to spatial variations. So the goal of my thesis is to improve spatial LDA to identify partitions due to both spatial and non-spatial variations. The methodologies includes clustering to find the multiple partitions in an image, applying spatial LDA to determine topics, and using spatial promiscuity to link partitions to LDA topics. And then we will examine this through a case study image. So now let's look at data set used in the thesis. The triple negative breast cancer TNBC dataset is by Carey, and this contains 39 TNBC patient samples and subsequent expressions details for 36 proteins. This data includes variables like sample ID, patient ID, which correspond to the clinical image, and each sample includes data on cells, their types, and the coordinates for cell centroids. And here we visualize the cell type distribution within samples. The heat map shows each sample as a row and each cell type as a column, with color indicating cell types frequencies. This helps us understand which cell type are present and their occurrences. However, this visualization does not show the spectral proximity of cell type. So meaning we cannot determine whether other cells are close to or far from the tumor cells. Incorporate spatial data. I have mapped out two samples, patient 4 and patient 12. Different colors represent different cell types, and with 16 cell types, the diagram become very complex and really hard to interpret. So hence we look for smaller or more perceptual partitions. We employed a spatial topic model where a topic is a layer structure consisting of a mixture of cell types and is described by a multinomial distributions. So well, we know that topic modeling is common for the textual data, and it can also help us to understand the complex MIBI data to capture the tumor microenvironment. So we treat all samples as coppers and partitions in each sample plus spatial coordinates as target events, cell types as term, and individual cells as words. And then for assumptions, LDA assume each partition is a mixture of topic and each topic is a mixture of cell types. In spatial LDA, the mixture of topic depends on the spatial proximity of partitions. And the following here's are some notations for my subsequent explanations. Okay, so now I'll talk about preparing data for the spatial LDA. We divided each samples into partitions using k-mean clustering, and then the plot shows the partitions for an image of patient 4, and we then compare the data frame from clustering result. Each row here is the partition, and with the centroid noted as LD and coordinates X and Y. And there is also a cell type count table for each partition showing the frequencies of each cell type in those partitions, as example is provided here. After clustering, we transform the result into long data. Each row now represents individual cells with its sample ID, assigned partition, cell type, and unique cell type ID. And we then consider spectral proximity between topics and partitions. LDA results given us the probability of each cell belongs to topics, and we create a new variable to assign cells to the topic with the highest probability. So then to find the center of the topic, we use the mean of the coordinates of all cells belongs to that topic and noted as LT. The centroid of partitions is given by the clustering. So we just get the spectral influences. We compute the distance between the partition centroid and the topic centroid, and use the normalized 1/distance as the spatial influence. And in the generative process of spatial LDA, for each partition D, we choose the topic proportion theta D follow the Dirichlet alpha, where alpha is given by the spatial influences here. And for each cell in a partition, we choose a topic and generate the cell type based on the topic's distribution. So this method accounts for both spatial and non-spatial variation in the data. Here is the topic proportion without considering the spatial proximity, and this is with considering the spatial proximity. So we can see that with the spatial proximity, we can capture the structure much better, especially in topic 4 and also in topic 3 and topic 5 too. Okay, so this slide shows two key Hamiltonian multicolor diagnostic. Left plot is the R-hat statistic indicating chain convergence, and we see all values are below 1.008, which is near 1, and it shows good convergence. And on the right plot is the effective sample size, ESS, indicating sample quality. A peak is around 2,000. That suggests a reliable parameter estimates. And in assessing our spatial LDA model, we compare blue histogram, which is from the model simulation, with the vertical line representing the observed cell count. And a different plot is for different cell type. The alignment of these vertical lines with the histogram peaks indicates that our model fit the actual data very well. And then we compare the fit of spatial LDA to standard LDA using the root mean squared error, RMSE, for each cell type. The scatter point shows most points above the blue line with slope of y. So it indicates that spatial LDA has a lower RMSE, and therefore it offers a better fit. So in conclusion, enhancing spatial LDA allows for better identifying the partitions due to the both spatial and non-spatial variations. And here are my reference, and thank you for your listening.
I do not believe we have them available online for questions, but they might be later on in the Slack for the conference Slack. So if you do have questions, by all means, shoot it out there, and maybe one of the two authors can respond. With that, we will start our next break. Please be back into your appropriate session. I think it's right here for lightning talks in 10 minutes.